bilateral side in green that are on the shorter branches. And it not only affects the sort of the length of the barbs, but it's really more the angle at which they insert into the rachis. And so what he decided to do is to try and look and see what genes might be controlling this. And so he did some transcriptional profiling, and he decided to look at basically at the wide veins versus the narrow veins. And so if we look here at the, he's looking at the lateral pulp versus the medial pulp, and he finds the medial one is wider, and so you wind up getting like crab binding protein, which is involved in bringing retinoic acids into the cells, whereas the CYP26B1 is over here, is higher in the lateral vein. And so you want to get a gradient, you want to getting a gradient of retinoic acid that develops across the feather. And he also found that gremlin-1 and follistatin inhibitors of BMP are upregulated over on the lateral side, and whereas GDF10 and FGF10 seem to be up on the medial side. And then he looked at the dorsal feathers, which have a very narrow vein, and the breast feathers, which have a wider vein, and he found the same molecules. So comparing the narrow vein to the wide vein is a similar kind of an effect. So then he wanted to show where they were expressed, and so keratin 75 is a marker of the rachis, that central backbone, and so it's expressed in the epidermis. And as we come down, these are primary images, which are the bilaterally asymmetric feathers, and then the secondary images have a certain amount of, a slight amount of bilateral asymmetry, not so much. The breast plume is bilaterally asymmetric, and the dorsal plume is narrower and has bilateral symmetry. And you can see that the size of the rachis is greatly reduced. And when you looked at the molecule that was upregulated, you found that GDF10 is expressed in the dermis beneath where the rachis is going to form. And so in each case, and the size of the GDF10 expression controls the size of where the size of the rachis that's going to form. He then looked at GREM1, which also was expressed, and if you look at this, he found regulates the barb generative zone, which on symmetric feathers winds up being directly opposite from the rachis. So that's where new feathers start to form, and then they come around and they join in to the rachis as they become a barb. So they start to form over here, and they go equally on either side to form barbs on a symmetric, bilaterally symmetric feather. But as you get up here to a bilaterally asymmetric feather, the position of GREM1 is shifted, so now it's going to be much shorter here on the lateral side than it would be on the medial side. And then when he was looking at molecules involved in setting up the retinoic acid gradient, he finds that the enzyme that digests retinoic acid is expressed somewhat equally on the dorsal plume, and as you move up more towards these, towards the primary midges, which are the asymmetric feathers, you wind up having the enzyme over on this side that degrades it, whereas the protein that helps to get it into the cell on the right-hand side. And so you wind up with this gradient. So in order to functionally test this, first he misexpressed GREM1, and he found that rather than having this nice feather shape, everything is much narrower width. And then when he started to look and see what was affected, he looked, first you can see the vein width is greatly affected. The angle at which the barbs inserted through the rachis also were diminished and were flattened. But the length of the growth of each individual barb wasn't affected. And then we looked at doing a similar thing with retinoic acid. He expressed the dominant negative retinoic acid receptor beta. He also got a narrower vein than he would have, or than you would have normally up here. And similar to what we saw with GREM1 expression, you saw that the vein width was decreased and the barb rachis angle insertion point was, or insertion angle was affected, but the lengths of the barbs were not. So using this sort of a strategy, we and other labs have been able to look at a number of molecules that affect the shape and the size of feathers. And so we found that BMP and sonic hedgehog are involved in barb branching, BMP and GDF10 are involved in determining the position and the size of the rachis, and Win3A also is helping to establish the position of the rachis. Then there is GREM1, as Angus showed affects the barb generative zone. Sprouting and FGF are involved in determining the size of the feather collar, and also branching along the proximal distal axis. And then there's retinoic acid gradient I just talked about is helping to establish the asymmetric feather vein. So if we look at feather, it initiates up at the top, and then it grows in about 25, 50, 75, and 100% growth. And we've looked at where different molecules are expressed that affect this growth along the way. And so during initiation phase, we find that FGF levels are very high, and as we move on down towards the base of the feather, its levels fall dramatically. And then BMP isn't quite expressed during initiation, but it comes up in early growth, and then its level increases as we get to the bottom. And some of these other molecules, Sprouty and Noggin, which affect sort of the width here, seem to be more highly expressed as we come out to the widest part of the feather, and then greatly diminished. And so working with Qing Ni and Seth Figueroa at UC Irvine, who are doing mathematical biology, they helped us to come up with a model for what we see in this particular case. So we think that we've got GDF10 that helps to establish where the rachis is going to be, and GREM1 that's going to help to establish where the bark zone is. And then GDF10 seems to affect this Wnt signaling gradient, and then that inhibits GREM1 to help to establish where GREM1 is going to be placed down towards the bottom over here as opposed towards the top. And then we also have some of the medial lateral retinoic acid signaling that comes in that also then feeds back to position where these signaling molecules are going to be. And that helps to establish whether it's going to be a radially symmetric feather, a bilaterally symmetric feather, or a bilaterally asymmetric feather. So another aspect of shape seems to be affected by hormones or sex. If we look at feathers, I showed you earlier that we do see these differences in sexual dimorphisms with feathers. And we were wondering if we took a body feather as a sort of a default type, and if we added estrogen, if it might look more female, if we added androgen, if it might be behave or appear more to be like a male feather type. Then we also wondered, well, if we take a male one and we expose it to estrogen, will it become female, and vice versa, a female exposed to androgen would become more male type. And Michael Clinton at the Roslin Institute had done a study several years ago where he was looking at a friend of his who was in the farm, owned a farm, found this one unusual bird which had, actually it's gynandromorph, it's female on this side and it's male on this side. And so even though it looks like a nice photoshopped image, it's actually a real bird. And so on this side you can see that it has, well, the comb is shared, so it's large. The wall is much larger on this side than on this side. It has more of a male patterning of coloring on this side than on the other side. And it also has a spur down here. And the whole body size is just larger. And when you look, in humans, males are XY and females are XX, but it's opposite in the avian world. Females are the heterogametic sex. And so he was able to, based on chromosomes, he was able to look and see what the sex of the different cells were, and he found the majority of cells on the male side were male, although not all of them. The majority of cells on the female side were female, although certainly not all of them. And so this led us to wonder how this other element might come into play, although we weren't working with gynandromorphs, so we don't believe we had to deal with something that might have both sexes. But again, the idea is that will estrogen make these feathers become more female, and will androgen make them become more male. And so the way we approached this was to do a gonadectomy study. And so when we did that, we measured hormones for 30 weeks, and over this time period, we saw that for males, testosterone levels were very high. And females, during the whole course of the study, were very low, as were all the ones that went through the gonadectomy. Whereas in females, the estradiol levels were very high. They were low in males, as well as all the birds that went through surgery. And so if we were to take a male and do a castration, we would find that we would be reducing the normal androgen levels, or testosterone levels. And if we did a similar kind of thing with a female, we would be reducing estradiol levels. And so we use that to try and understand what's going on with feathering and also with the cones and models. So this is a normal male, and you can see that it has a large comb and large wattle, and it also has this big the tail feathers coming out the back. Whereas the female has a smaller comb and wattle, and it has much smaller feathers. So in our males in the food industry, they generally will do castrations around 2 weeks of age, and that's to make capons, which are they're a much more gentle bird for people who have small farms, and they also tend to have more tender meat, so that's why they do it there. And here on these birds, they still have the same male tail feathering, although their combs and their wattles are greatly reduced. So it seems that androgens seem to be involved in affecting the combs and wattles, but it seems like it's more a lack of estradiol that may be affecting the types of feathering here on the back. And also, if we take the females, after doing the gonadectomy, we find that they start to grow male feathering. So, and I'm not quite sure what to make of that their combs and wattles are much larger than they are normally. But, so there's got to be some cross-talking on these hormones as well. We have to look into that further. So when we started to look at the hackle and the saddle feathers, just the shape of them, we can see that the hackle feathers have sort of a tapered tip as we go up here, and so do the saddle feathers in normal males. And in the normal females, they're much broader feathers, both for the hackles and the saddles. And then when we looked at the castrated males, they retained mostly their original feather shape. So it didn't seem to be affected by a loss of androgens, rather it was a lack of estradiol that seemed to be the effect, that seemed to be implied here. And for the females, when they lost estradiol, they assumed the male sort of so that's also consistent. So in summary, we can show that cells are able to self-organize and form different appendage structures, and that feather shape and diversity can be regulated by GPF10, which positions the rachis, as well as retinoic acid, forms a gradient that can establish this medial-lateral axis determination, and it can position GREM1 and the progenitor zone. And then also that hormones can guide feather morphology and diversity, and that it seems like in our particular case that estradiol is the one that keeps, that will change the feather shape rather than testosterone. So I'd just like to acknowledge that the work that was done on feather shaping was done by Ang Lee in the lab, and the work that was done on the hormones was done by Pinchi here in the lab as well. And these are the other lab members who also contributed to everything. And I want to thank our funding from the NIAMS. So I'd be happy to take some questions.
Thank you very much for your clear and very interesting story. Do anyone have any questions or comments?
Oh, that was a spectacular talk. Thank you, Randall. So this plasticity of the feather in adult birds is quite amazing. How much of that is still conserved in the mating species?
Well, well, first of all, it seems to be different in terms of androgen seems to be the more controlling factor in mammalian species, and I don't know if that's because of the difference in the heterogametic sex, whatever. But I don't know that many people have really started to. I haven't seen a lot of studies where people have gone and played around.
But there are clear observations. You see that after puberty, the hair shape can change, sure. And then after chemotherapy, regrowing hair can have a completely different change than it had before. So we must have retained some plasticity, but I think none of us really knows what controls it. Well, from you guys, no?
Yeah, I mean, there are certainly, there are those type of changes that you see, and it's a matter of nobody really looked into so much what kind of things are regulating.
But since you've mentioned so many different factors, we couldn't possibly study all of that now in a human follicle, right? So if we have to start somewhere, where would you, if you would have to put all your eggs into one basket, which factors should we start to look at first? Human, not mouse.
No, I understand. Well, I mean, some of the things that we're doing, like with the hormones and stuff like that, I mean, people are doing those kind of studies, I think, in humans as well already. Some of the things that are affecting feather shape, I'm not sure how relevant they're going to be to the shaping.
But the molecules are all the same, right? So
that's true, yeah. I mean.
So you can't give us any specific recommendation. That's what I'm trying to explain.
Well, I understand. I mean, certainly there's not a racus or anything, so we can't look at the things or the pore genders or so. But some of the things, the retinoic acid gradient that helps to establish some kind of a gradient or whatever may be common in the
Margaret Hardy did that, right? In the mouse, right? Ages ago.
Yeah.
Okay. Yeah, I shut up.
Anyone else?
Okay, so our time schedule today is very tight, so I would like to move on the next speaker.